Hello everyone and welcome to week 8 of NPTEL course in Herbal Drug Technology and Regulatory Sciences. In this week we will see different regulatory requirements for a pharmaceutical drug manufacturing, particularly in the herbal scenario. In this particular session, let's focus on pharmacopoeias, the monographs as well as the books on quality standards. So let's first understand a book called as Pharmacopoeias. Now what are pharmacopoeias? Pharmacopoeias actually derive their name from a Greek word pharmakon, which definitely means medicine, and a process to make which goes as peon. So pharmacopoeia is a process of making a medication. This arises because if you see the earlier volumes of any pharmacopoeia, that is this official books, you will see different methods of preparations of medicinal formulations. So how to make this medicinal formulations were devised and stored in national books, and these books, depending upon their countries, were called as respective pharmacopoeias. Say for example, in India we call it as Indian Pharmacopoeia. For US you will call it US Pharmacopoeia. There is British Pharmacopoeia, Japanese Pharmacopoeia, and so on. So most of the countries have their own pharmacopoeias, which in the earlier days documented the official recipes of different formulations, and later on went into kind of categorize it as drugs containing monograph. Later on went to categorize it into subcategories where formulations became one volume of it, and the APIs, that is the active pharmaceutical ingredients, as well as the excipients, became another volume. Gradually over a period of time, as the drugs increase, most of the pharmacopoeias now focus on drugs. So what basically is pharmacopoeia? If I have to define pharmacopoeia, it's an official book released by the government or by the ministry. It's legally recognized and set standards for quality, purity, strength, and if I have to perform analysis or quality control of any medications, particularly API, it will also give me the process of quality control or process of testing, which is used or pertinent to that country or region. So if I have to say Indian Pharmacopoeia, it will be legally binding to the Indian geography, and most of the preparations found herein as well as the drugs that are prepared herein have to be Indian Pharmacopoeia compatible. So this compatibility is somewhere documented in form of a monograph which contains detail of a drug. So like every book has a chapter, a pharmacopoeia has a monograph. Say for example, for drugs like aspirin, it will have an aspirin monograph. For herbal drugs like acacia, for herbal drugs like your castor, or for herbal drugs like your datura, it will have a monograph of a datura. So similarly, now when you say it is a legal document, the country, especially all the manufacturing units in a country, have to be compliant. So that's a legal requirement. So if I have to manufacture, sell, import my drug in India, it has to be Indian Pharmacopoeia compliant. Only then it has to, or only then it can be manufactured, it can be sold, it can be imported. So country wise, if you go to see, there are different pharmacopoeias. For India, there is Indian Pharmacopoeia, then you have your British Pharmacopoeia, European Pharmacopoeia for the whole European Union, USP, Japanese, Korean, Chinese Pharmacopoeia. These are the country wise pharmacopoeias. Now you also have pharmacopoeias based on systems of medicine. So if you go to see Indian system of medicine like your Ayurveda, we have the Ayurvedic Pharmacopoeia of India, we have the Siddha Pharmacopoeia as well. So pharmacopoeias are legal books wherein you see different parts of pharmacopoeia giving details of drugs, their specific requirements, in some cases formulations, as well as their testing protocols. So, what are the objectives of pharmacopoeia? Why are these books written? Now these books are written and mandated by government. There is a reason for it. The basic reason is to protect public health. So if there are no rules governing how a particular API, or that is active pharmaceutical ingredient, or how a particular herb should be at the time of manufacturing as well as in its finished state, that poses a public health risk. Definitely there is going to be adulteration if there is any spurious or substandard drug that need to be detected early in order to ensure public health. And in order to do this, it has to be Indian Pharmacopoeia compliant. That means any drug or any herb that meets the IP requirement would definitely be free of adulteration, substitution, or any other spurious ways. So it protects public health by establishing a very authoritative standards which all the industries should be compatible with. Not only that, it uniforms, or it kind of lays down a very uniform standards. So no matter how big or how small the industry is, there is no excuse to quality, and all the industries have to prepare their ingredients and see to it that the raw materials, especially the APIs as well as the herbs as well as the herbal products, are of uniform quality. Not only just the drugs, it also ensures, or it also contains monographs, like chapters of excipients, which are your inert material that are added to the formulations and that also are ensured of its quality. Not only that, the Pharmacopoeia is a book which lays down regulatory control in a way that tomorrow if any authority finds that my senna as a drug is of substandard quality, it's not complying with the Indian pharmacopoeial standards, I have the complete license to reject that consignment of senna and seize it because it is not compliant for quality standards to be produced, manufactured or sold in India. So definitely with that quality, what you call it as enforcement, I somewhere ensure safety of my patients. Not only that, the objective of pharmacopoeia is also to promote rational drug use, a good pharmaceutical quality assurance. Now this pharmacopoeias, depending upon country to country, regularly update. So if you see Indian pharmacopoeia, maybe somewhere deep down the first volume of Indian Pharmacopoeia came in as early as 1955. During those days, we didn't have advanced techniques like HPLC, gas chromatography, HPTLC, which were very accessible to the industries as well as personnel. So then the quality control methods were very simpler. But as we move down to Indian Pharmacopoeia volume IP2, that is the 10th edition, IP 2026, here more sophisticated techniques are used. So pharmacopoeias as books are continuously updated by the government to match, ensure the quality and compatibility, so that something that is IP compliant would also be of quality that industry could export it. So it harmonizes with other countries to ensure similar quality control methods as well as regulatory as well as regularly updates on the same. So the objective of pharmacopoeia is public health and interest at first, laying down a very strict quality control guidelines, a rational drug use, regular updation as well as harmonization with other countries. Now, what does the pharmacopoeia contain? Now, if I open a pharmacopoeia, I get information about drug. This drugs includes herbal drugs. This drugs include vaccines, biologicals, active pharmaceutical ingredients which come out of synthetic origin, herbal origin, metal, mineral origin, etc. So whatever are drug ingredients as well as excipients, many of them have pharmacopoeial mentions. So much so that if you see the Indian pharmacopoeia in the current scenario, there are more than 3,500 monographs of drugs, excipients, biologics, vaccines, etc. so that quality of those products in market is ensured. Not only that, if you see the earlier volumes as well as certain pharmacopoeias like your Ayurvedic pharmacopoeias, they also give you method of preparation of formulas. So if I know a formula called as Triphala Churna, the Ayurvedic pharmacopoeia will give you a precise method, quantities as well as method of assessment of my Triphala Churna. So method of preparation of formulation, quality control methods for my actives, my excipients as well as my formulations. Now in some cases it also gives me details of reference compounds. Now what happens is, say for example, tomorrow in my industry I am preparing Senocide or I am preparing aspirin. Now how do I compare it with? So there are certain compounds which are called as reference substances. These reference substances are substances of known purity, the purity of which is assured by the government, and they lie in the repositories with the government as reference substances. The spectroscopic, the analytical data of this compound is available, and sometimes such compounds analytical data is also the part of pharmacopoeial content. In addition to that, it contains general information about the solubility, about the general methods of storage, handling, sampling and so on, as well as the appendices which mentions over a period of time how to do the quality control methods. The processes, the procedures and the amendments are also part of the Indian pharmacopoeia as well most of the pharmacopoeial books. Now if you go to see monograph, monograph is what we call it as the heart of a pharmacopoeia. The monograph is a chapter. So it's a detailed written document. It's an official document which sets what are called as quality standards. It gives us what to carry out in terms of test. What is the acceptance criteria? What is the limit? Say for example, if I am talking about amla, how much amount of tannins it should contain for me to be acceptable. It might so happen that the amla powder that I am handling is substandard, it's already extracted and exhausted of its tannin such that it contains 0 tannins. So if my pharmacopoeia says that it should at least contain 1% gallic acid, when I analyze it, if it is containing less than that, that means I have to reject that sample. I cannot use that sample for my manufacturing, neither I can use it for my sales or any processes pertaining to the pharmaceutical industry, especially in cases of herbal medicines as well. Now it will give you specific tests. It will give you tests for identity, strength, purity, efficacy, and these are something which are required to ensure consistency and safety. So only when all of these tests are accurate, all they are done and they are compliant with the pharmacopoeial limits, you can say your drug is safe and efficacious to use. So the monograph is a written document which acts as standards, which contains specifications, which contains analytical procedures, requirement of physical, chemical properties, dosages, packaging, how to store it and what is to be written on the label is all a part of monograph. So what are different types of monograph? So depending upon the contents, there are different type of monograph. Generally what we see in Indian pharmacopoeia is what is referred to as standards monograph. Now when I am talking about standards monograph is more to do with the physical and chemical attributes. So all the physical as well as chemical properties of a herbal monograph or of a herbal drug are seen in standards monograph. Whereas when I refer to therapeutic monograph, it is more to do with the biological activity. So standards monograph sets up quality. Therapeutic monograph is more often used to ensure biological activity as well as efficacy. So in some cases you might find a combined monograph, which is a combination of standard and a therapeutic monograph. So let's see the details of a standard monograph. So when we are talking about standard monograph, like I said, the standard monograph deals with the physical and the chemical properties. Now it could be the physical and the chemical properties of a herb, physical and chemical properties of a substance which might be an unorganized drug, or physical and chemical properties of an excipient. So in terms of its parameters, we generally see parameters to ensure identity. How do I identify this drug? Potency, that is what is the strength of this drug. Purity, how much pure is this drug? So this is all explained in the standard monograph. So standard monograph typically includes a layout which gives details such as name, chemical formula, physical properties, solubility of the compounds, official tests, like I said, maybe some IR, NMR, spectras, list of impurities, test for impurities, acceptance criteria based on this limit test. So a good example of standards monograph is your Indian Pharmacopoeia monograph. So let's see an example of an Indian Pharmacopoeia monograph and try to understand this. So what you have here is an example which is taken, or a sample which is taken from Indian Pharmacopoeia, and this is a monograph of acacia. So in Indian Pharmacopoeia you again have monographs of allopathic as well as herbals as well as biologicals. Since we are doing herbal drug technology, let's study your pharmacopoeia, that is the monograph, with respect to herbal drugs. So acacia here is an example of excipient. Now pertaining to Indian Pharmacopoeia, you will see that whenever there is a excipient involved, the monographs slightly differ from a monograph of drug. How is that? Let's study that as well. So when I'm talking about the monograph, as you can see here, you will have your name of the drug or name of the excipient, which is your acacia, the common names, then a scalar image. So this scale is generally a 1 centimeter scale. So this is like 12 centimeter by 10 centimeter square, and few what are called as tiers of acacia with vivid color are placed in. So this will tell you about the appearance, about the description. So you can see the biological sources from which this gum is obtained. Then you will see the category, which is pharmaceutical aid and in some cases anti-diabetic. Description, how it occurs in market. So in terms of description, you can see that it occurs in both forms. You can purchase it in form of the whole tiers, or you can purchase it in form of powders as well. The description of powders is given. Then you might come across numerous such unorganized drugs in market. So how do you identify that this is acacia and this is not any other gum like your tragakant, karaya, agar agar and so on? So it will give you what are called as test for identification. So these are generally the chemical reactions. So here it is like gelatinized by lead subacetate, as well as you can check with ammonium oxalate. And if you see here, the 10% solution may show a dextrorotatory as well as slightly levorotatory activity. So you can even check the identity in terms of optical rotation. Then it is followed by tests. Now this tests are to ensure that it is free from adulteration. So if I see, or if I have acacia as a drug and then it is adulterated by some other substances. Now this other substances might be sterculia gum in this case, you can see here. So for sterculia gum, it gives you a chemical test like treat it with ruthenium red, and since it's not mucilaginous, your agar should not turn red. Similarly, You can see a good quality acacia should not contain agar or tragacanth. You can check that by lead acetate test. It should not contain starch or dextrin as an adulterant. Just add a drop of iodine and you can see. It should not contain tannins, which you can detect by a simple ferric chloride test. Then it should not contain sugar powder. Like I said, there are different adulterants in market and your IP ensures that they should not be there by just doing simple chemical tests. So sucrose and fructose, you can simply do a Selvinov's test. Then you can check for what is called as water insoluble matter. Now this acacia gum, when it is hydrolyzed by hydrochloric acid, should get converted into its corresponding sugars and should easily flow through any filter paper. Now if it contains any insoluble matter or particles, that is called as water insoluble particles. So that is checked and the limits for that is also laid down. Then you have microbial contamination. So this being used as a drug as well as an excipient, you will see it should be free from E. coli, which might be pathogenic. Then it gives you physical methods of evaluation. This includes your ash values. You remember, this ash values include the sulfated ash. This is generally done because this is more of an unorganized drug. Then you have acid insoluble ash to take care of silica and other non-physiological ash material. Loss on drying, which will tell us about the moisture content as well as the volatile oil content. Now if you see this monograph in detail, you will see in every case there is a limit which is laid down. So when I am saying loss on drying, it should be not more than 15%. That means if there is a moisture which is more than 15%, there is a high chance this drug will deteriorate due to microbial or other contamination and hence it is not acceptable. So a moisture content or a loss on drying should be less than 15%. Storage, how do you store it? Now I will see an example of a drug. So in this case, since it's an excipient, you don't have what is called as assay. So in the next drug where you have amla, you can see again you have a drug name, you have a description, you have a scalar image, biological source, the category, that is the therapeutic category, description, then you have identification test. Here because it's a drug, you can see a little modern method like your TLC is being involved for checking gallic acid and tannins, wherein the mobile phase is also given and method of preparation. Then post that, if you move to this side, post your TLC, you have your physical method of evaluation, which includes your foreign organic matter, extractive values like ethanol soluble, water soluble, ash values. Here you check for heavy metals. So limit test for heavy metals is to be done. Loss on drying. Here you see earlier it was 15%, for amla it is 12%. So these limits are not generalized, they vary from drug to drug. Microbial contamination, and then this being a drug you also do an assay. And IP, this is IP 2018, which includes a HPLC method for determination of your gallic acid, and the last part is storage. So you have to store it away from insects and rodents. In terms of HPLC, the content should be not less than 1%. So that is how your Indian pharmacopoeal monograph goes, and that is what is called as your standards monograph. Now we go to the second category of monograph, and second category of monograph is your therapeutic monograph. Now the first one ensured identity and quality. The second category of monograph, that is your therapeutic monograph, mostly focuses on bioactivity, safety and efficacy. So therapeutic monograph do deal with more of your pharmacokinetics and dynamic parameters. So therapeutic monograph definitely give you a little bit about detail about identity, but it's more focused on efficacy, safety, toxicity, detailed pharmacology, clinical evidence, if there are any contraindications, adverse effects, warnings, those are also seen. A good example of such kind of monographs are what are called as your ESCOPE monograph. Now moving on to the next category, the next we have combined monograph. What if we have best of two? The standards monograph, which will tell me about the quality, as well as a therapeutic monograph, which will give you about the bioefficacy, safety and contraindications. So such monographs are referred to as combined monograph. A combined or a combination monograph has both of this, and you will see such in your WHO, that is World Health Organization monograph, as well as you can see this in your American Herbal Pharmacopoeia monograph. So let's see examples of these two monographs and try to understand what added advantages do combined monographs have over standard as well as therapeutic monographs. So just giving you examples with WHO monograph first and thereafter your American. So here what you see on screen now is an example of WHO monograph. It's a simple monograph of garlic, that is your Allium sativum. So if you see as compared to the standard monograph we saw in the Indian Herbal Pharmacopoeia or the Indian Pharmacopoeia, you see here it has name, botanical name, synonym, vernacular name. Similar way your microscopic description, appearance, organoleptic microscopic properties, powdered plant material. Now a add-on which was not there in your Indian Pharmacopoeia, here you will see your geographical distribution because it is WHO monograph. So a little bit of geographical distribution is also explained, identical test, and you can see it's very similar to your IP monograph. Identity, purity, microbiology, physical methods of evaluation which includes ash value, that is total acid insoluble ash, water soluble, alcohol soluble, extractive values, water soluble extractive values, moisture content. It also checks for pesticidal residues, heavy metals. Because it's global, you also have to check for radioactive residues if there are any in the drug. Now other chemical assays, it gives you in-depth knowledge about different chemical constituents and their stability with structures which are present in garlic. Dosage forms, now you will see you are moving from a standard monograph to a clinical or a therapeutic monograph. So you can see different dosage forms, medicinal uses where all it is used, which all traditional systems of medicine it is used, experimental pharmacology, detail of how it works. You can see with respect to the papers, with respect to the mechanism of action, that all is also there in your WHO monograph. And if you go further, you will see different clinical pharmacological evidences of clinical trials, meta-analysis, and then this is followed by your contraindications, warning, drug interactions if any that are observed, effects on pregnancy, nursing mothers, any other precautions that are to be taken, adverse reactions, posology as well as references. So if you see as compared to your Indian Pharmacopoeia, this has much depth of knowledge. The best sample if I have to give you of a combined monograph, it would be of your American Herbal Pharmacopoeia monograph. So let's study this monograph in detail again. So here I have taken one monograph and that is from American Herbal Pharmacopoeia and that is your echinacea. Now just see the comparison as compared to the previous one. We saw the Indian one, we saw the WHO one, and you can see American Herbal Pharmacopoeia how much they are reliant on quality parameters. So you can see your herb with all the documented evidences. Here the contributors are also cited, which we did not see earlier herein. So you can see a great credit is given to the contributors. Now the actual monograph starts with botanical nomenclature, family, definition, common name, and add-on which also is there is your history. A complete botanical identification, geographical identification is seen in the monograph. So you can see the dried, fresh flowers at different stages of blooming. Then you have different photographs of microscopic and macroscopic. Not only that, you have the details of allied species also. So if there are any different petal colors, if there are any different variants which are less or greater in height, all this are being documented very properly in the American Herbal Pharmacopoeia. When it comes to microscopy, it is supported by lot of microscopic images as well as tracings, which are, these tracings are to the scale and very exact. So inflorescence, flowers, fruits, the kind of stomata, the kind of trichomes, the kind of pollens, everything is documented by the American Herbal Pharmacopoeia. This includes your microscopy, where are the commercial sources, how do the commercial sources appear, their structure. So again, the contents in terms of the chemical composition. So here if you see, the chemical composition is just not specified randomly. You can see the chemical composition here is specified depending upon different plant parts also. So, you know, as Indian Pharmacopoeia or as different pharmacopoeias, we take aspiration to get to this depth so that we can ensure absolute quality of substances. The cultivation, the collection techniques, the collection season, everything is documented here. The post harvest, how it is to be dried, how it is to be processed, you get a very detailed knowledge on that. So that's why these monographs are very elaborative in nature and go for pages altogether. So complete knowledge of any plant can be obtained. So in terms of constituents, you can see there are many pages of chemical constituents with structures, the yield, the compounds. It's just not like one compound which is standardized. There are multiple compounds which are standardized herein. The HPTLC, the HPLC details, the samples, the images. Now these are nowadays also obtained in Indian Pharmacopoeia as well. So if you see Indian Pharmacopoeia, gradually the newer volumes contain the HPLC and HPTLC images. They have standardized it with respect to most of the compounds, the HPLC as well. Then you start with the therapeutic monograph. So in therapeutic monograph, you will come across different animal studies, pharmacokinetics, pharmacodynamics, clinical evidence, the dosage, contraindications, everything are much in detail. So you can see here, it's like a 64-page document just for one drug. That's the kind of documentation they do, and it's a very detailed monograph. Any adverse effects herein which are found in human volunteers, which was done on animals, are all documented in here. So you can come across dosages, adverse reactions, the safety profile, contraindications, overdose or any treatment which is available on overdose. So these are all the things, and you can visibly compare starting from Indian Pharmacopoeia, which is a much simpler standard monograph, to WHO, which is an example of combination monograph, to American Herbal Pharmacopoeia, which is a combination example but contains all of this in much detail. So these are made through crowdsourcing, public sourcing. They are made available, but definitely they are paid. Now seeing the Indian pharmacopoeial scenario with 2026 just on the horizon, we already have released our IP 2026 on 2nd of Jan, that is the 10th edition of Indian Pharmacopoeia. Now this Indian Pharmacopoeia is managed by Indian Pharmacopoeial Commission, which is a body under the Ministry of Health and Family Welfare, which is Government of India body. Now this is a legal mandate, and anything that is to be manufactured in India, imported, sold or distributed in India under the Drugs and Cosmetics Act 1940 and the Rules 1945 has to be all compliant with this IP. So apart from this, we also have Ayurvedic Pharmacopoeia. Just wanted to show you that monograph as well. So if you see the Ayurvedic Pharmacopoeia monograph, it again gives you much in detail about the drug, which is the same drug, Amla I have taken here. You can see the description, again identification in terms of physical values, but in addition to that you also have a little Ayurvedic properties and actions to it. So this tab is a little extra tab that you see. Now in terms of its therapeutic uses, it would be more of an Ayurvedic therapeutic applications, as well as the dose is mentioned. Now in addition to that, you also have IP volume 2, which gives you the detail of formulation. So you have drugs, you have Ayurvedic formulation, same applies to your Siddha and other systems pharmacopoeia as well. So here are few references. So what we studied so far are your different pharmacopoeias. What is pharmacopoeia? The objective of pharmacopoeia, different types of monographs. You saw the monograph in Indian Pharmacopoeia, you saw a monograph of WHO, American Herbal, as well as an example of a systems pharmacopoeia, that is your Ayurvedic Pharmacopoeia of India. So thank you everyone for your patient listening.